Well, thank you so much, Lee, having organized this meeting, for bringing me here. It's a great pleasure to be back at MIT. And yeah, I will be talking about relations, the problem of encoding, retrieving, and representing relations in the brain. Why relations? Because they really matter. Cognitive objects, they are defined by features and the relations among the features. This is what is constitutive for them. And therefore, all cognitive systems need efficient mechanisms to detect, encode, and classify relations if they want to establish valid models of the world surrounding them. Now, all relations are defined, all meaningful relations are defined by statistical contingencies in space or in time. And therefore, mechanisms that evaluate relations, they need to compute correlations. This is a general thing. And there are two strategies that nervous systems seem to exploit in order to detect and encode relations. One is the straight feed-forward generation of conjunction-specific neurons, where feature-detecting neurons converge onto higher-order neurons that reconverge onto yet higher-order neurons until one ends up, if the weights here are adjusted appropriately, with conjunction-specific neurons that respond only and only if a particular set of input constellations is present. Because these neurons always respond to the same constellation of features, we call this the labeled line code. Such a neuron stands for a particular relational construct. The complementary alternative, which is also applied, is the good, well-known Hebbian assembly. Neurons get associated ad hoc on the fly to form a functionally coherent assembly, and the features that are represented by these individual neurons for that moment of time are bound together, and the assembly stands for the representation of a relational construct. In most nervous systems, in particular simple nervous systems, but also in the now so much discussed AI systems, the deep learning networks, the feed-forward binding of features is the optional strategy. Conjunction-specific neurons are generated by weighted convergence of input neurons over several steps of processing. Very efficient strategy, very economical strategy, very rapid strategy, but it has disadvantages. There are no memory functions in such networks, so it is difficult to superimpose subsequent states, which makes it difficult for these networks to handle sequences. Also, these networks have no self-organizing capacity. They are essentially linear feed-forward structures. So, in both situations, in both solutions, relations have ultimately to be recoded into temporal correlations. One reason is that the world is just working like that. Events that are simultaneous tend to be related. If A precedes consistently B, A seems very often is the cause of B. So it makes sense to extract temporal correlations from the environment in order to detect relations. But also, and because of that probably, all the mechanisms of synaptic plasticity, they are exquisitely time sensitive. They evaluate associations with the precision in the millisecond range, be it spike timing dependent plasticity or other forms of use dependent synaptic plasticity. They all evaluate temporal contingencies. So eventually both labeled line codes and assembly codes have to exploit temporal contiguity. If a labeled line code, if a conjunction-specific neuron were to be formed by experience, it needs to be assured that input from feature detector A and feature detector B be made temporally contiguous in order to exploit the plasticity rule that would enhance correlated input that is correlated in time. Now there are two sources of precise temporal relations among neuronal responses. One is straightforward, stimulus locking. If two events in the outside world occur at the same time, the responses will be synchronized in the brain, and we know that even across multisynaptic pathways, at least the transient sensory systems, the phasic ones, can transmit external temporal signatures with very high precision in the millisecond range over several synaptic stages. But there are also, of course, many situations where there is no temporal signature to representations that need to be associated. Like if one retrieves two contents from memory that should be bound together, they don't have a genuine temporal structure to begin with. So one way out is either to have a completely different set of plasticity rules that are not time sensitive, and to the best of my knowledge, they have not been discovered yet, or one has to generate internally temporal structure and align activity patterns that should be bound together in time. Now, oscillatory patterning is an ubiquitous solution in order to pass time, in order to impose temporal structure on neuronal response sequences. Even in very simple animals, invertebrates, we have pacemaker neurons, pacemaker currents, oscillatory neurons. They are associated together. They perform central pattern generators. We have this in the spinal cord, the respiratory. So oscillating circuits are ubiquitous in nervous systems, and they are used in order to pass time, impose temporal structure on self-generated activity. Now in the cerebral cortex, and we have heard a lot of these time generators this morning, there are abundant oscillatory microcircuits, which this is taken from Nancy. This is the ping circuit here, but we have heard today that there are many, many other possibilities to generate rhythms in different frequencies. It all depends, or most of these rhythm generators are based on recurrent inhibition, whereby the time constants of the neurons, the conduction times, determine the frequency of these oscillators. They are usually damped oscillators, which frequency of which depends on the excitatory drive and on modulatory signals that impinge on these microcircuits. An old, actually, the finding that got us going into this field of dynamics was that because of these microcircuits that exist in cortex, if one activates a bunch of neurons with the appropriate stimulus to which they can respond, they get entrained into these rhythmic oscillatory activity that is reflected in the oscillatory field potential. An ubiquitous phenomenon, easy to detect, and I think no longer doubted to exist. Now, once one has an oscillatory signal, once one has an oscillator, one can generate very precise temporal relations among sets of oscillators. And this can be achieved by reciprocal coupling. And we know that most of the columns that contain these oscillatory microcircuits are reciprocally coupled. And the outcome of this coupling is formulated by the theorem of the Arnold tongue. We have in the cerebral cortex a dominant wiring principle, and these are those red lines, these red fibers, which are recurrent collaterals of pyramidal cells, essentially in supra and infragranular layers, that cross tangentially in the cortex and connect in an excitatory to excitatory way neurons in adjacent and remote columns. Some of these connections also terminate on inhibitory interneurons, but what matters here is mainly this reciprocal excitatory coupling of these oscillatory microcircuits. The consequence of this coupling is that these columns can synchronize their activities if the coupling is strong enough and if the resonant frequency that they prefer is similar enough. The more the preferred frequencies of these oscillators differ from each other, the stronger the coupling has to be in order to still establish synchronization at various phase angles among those oscillators. With very similar resonance frequencies, very, very weak coupling is sufficient in order to entrain oscillators into synchrony, as has been shown by Van Huygens with his pendulum clocks that he one morning found all in synchrony because they were hanging on the same timber and the vibrations of the timber took care of the coupling. So this is what one finds, also an ubiquitous finding if one records from two oscillators not too far distant, activates them, they engage in synchronous oscillations. They can be zero phase lag if the coupling is reciprocal. So zero phase lag synchrony does not necessarily mean common input. It can also emerge from reciprocal interactions. And even with conduction delays up to 1/4 of the oscillation cycle, zero phase synchrony can be established by reciprocal coupling alone. Now, the question arises how semantic relations can be converted into temporal correlations. And one example I would like to give is from the visual cortex, where we know that the coupling strength of intercolumnar recurrent connections, of these synchronizing connections, actually reflects the statistical contiguity of features in the outside world. And these anisotropies, the weight distributions of these coupling connections, they correspond very well to the Gestalt rules that we use for perceptual grouping. And I will give you an example. The specificity of these tuned, natural, reciprocal, excitatory connections is to some extent genetically specified. There's an exponential distance rule, so the coupling among columns decreases exponentially with distance. That takes care of the grouping criteria of vicinity, the Gestalt criterion of vicinity. Columns that are nearby are more strongly coupled than columns that are far apart. But there is a massive epigenetic modification by experience of the layout of these connections on top of this distance rule. With the result that the weight distributions of these connections reflect in great detail the statistical contingencies of features, implementing the priors that are later used in the Bayesian matching process for scene segmentation, feature binding, and so forth. And here is an example. It's an old study, but still true, of how these connections are shaped by experience, implementing the statistical contingencies of the outside world into functional architecture. At that time, methods were still primitive. We mapped the functional organization of striate cortex, the columnar organization with deoxyglucose, not with optical recording. So these are orientation columns, then injected fluorescent beads and had these beads transported along the tangential connections to see how the network of these connections would be in register with the functional architecture of the cortex. And it turned out that in normally raised animals, this is cat visual cortex, there is a striking selectivity in the connections between functional domains. Neurons connecting very selectively, these are two different populations labeled, the same functional domains, in this case, domains sharing the same orientation preference as the injection site. This is very different in animals that are dark-reared, where the connections are more or less remaining random. So experience shapes the architecture of these connections to a substantial extent, implementing or representing statistical contingencies of features in the outside world in the network of these tangential connections. This will become important a little bit later again. So one can consider at least the supragranular layers of cortex as a recurrently coupled network whereby the coupling occurs with delay, which is important because that complicates the dynamics, makes it more interesting, increases dimensionality. It's anisotropic. Is a nearest, so individual columns can be considered as oscillatory microcircuits as feature-selective oscillators. They are coupled through this nearest neighbor rule with an exponential decay. This is genetically determined. But in addition, there are selective couplings that are shaped by experience that tend to couple more strongly oscillators that code for features whose co-occurrence has been examined in the world outside or that co-occur very often in the outside world because they are constitutive of certain objects and need to be bound together in perceptual grouping, for example. So, in this way, the Gestalt rules that reside in the functional architecture of these recurrent connections get translated into synchronization patterns because the Arnold-Tang theorem predicts that strongly coupled columns should synchronize with higher probability than weakly coupled columns. Ergo, if features occur, drive columns, if features occur that are often contingent, that should be bound together, drive their respective columns, these columns are already more strongly coupled than others, they will synchronize with higher probability, and thus synchronization probability eventually reflects Gestalt criteria that are used for perceptual grouping. Also, this is what the data show. If one records from two groups of neurons and drives them with a stimulus that is coherent with features that are usually bound together into a single object, the neurons synchronize because these groups are strongly coupled, sharing the same direction preference, while if they are activated by two discontinuous contours, even though they respond nicely, they don't synchronize. So far, so good. It seems as if the same principle were applied to interareal communication. So not only to bind together columns that code for certain features, but also at a much larger scale. And we have heard today beautiful illustrations that this seems to be the case also for the gating of communication by synchronization between different cortical areas. And it's clear that it is also a mechanism that is used for the formation of functional networks. Actually, we measure coherence in order to identify functional networks because we believe that coherence is somehow telling us something about the communication between these networks. And this seems to occur at the brain-wide scale, and it has already been mentioned this morning, and I will only be very brief here, that measures of synchrony are used as a diagnostic tool, both for the identification of functional networks, but also for the diagnosis of abnormalities in such networks. And we have heard this morning that there are problems in certain diseases, psychiatric diseases, in schizophrenia, but also in autism spectrum disorder and Alzheimer's, I learned this afternoon, that there is something wrong with the temporal coordination of distributed activity. And this can be measured easily by analyzing in human subjects, in controlled populations, populations affected by a disease, if subjects are engaged in this case, rather simple cognitive operations like detecting in this mood whether such a black and white contrast picture is a face or not, the so-called Mooney pictures, just to keep the subjects busy. Then record with MEG, make a time frequency analysis with wavelet analysis, and then source localize with beamforming, and then diagnose the dynamical responses in these cases. And what has been shown over and over again, and this is just one example, in this case, not the amplitude of oscillatory activity is measured, but the consistency of phase locking in controls after presentation of such Mooney phases. They have finally detected them. One finds these bursts of gamma activity with fairly precise phase locking across large distances in the cortical network, which is much less expressed in patients affected by the disease. It's also abnormal instance, in first admission patients, so it's not a medication artifact, it is a true sign of disease. And the correlation of this difference with positive symptoms is quite impressive when they are independently determined by clinical colleagues. Comparing at the source level, at the sensor level, at a particular time after presentation of these Mooney phases, the coherent values at a certain threshold level, this is what one sees in normal subjects. So this simple cognitive task engages a large array of cortical areas, which is much so in the schizophrenic patients at the same threshold level. More recently, we performed a more detailed analysis of functional networks at the source. And as one would expect, there are many networks affected. Differences are seen in many different networks. Because of the task employed here, it's of course a lot to be seen in the visual system, but also other network components of other networks, including the default network, are affected. So the conclusion from this line of research, which is done by many laboratories now, is that the power, the synchrony, and the phase locking of oscillations are disturbed in schizophrenia, also in autism spectrum disorder, and as we heard today, also in Alzheimer's. It's still an open question whether this disturbance in dynamic is causative for the dissociative symptoms in these diseases, but it can at least be used as an endophenotype. There is now evidence that one can also diagnose already at risk before they have manifest clinical symptoms on the basis of such disturbed dynamics. Now, I want to switch gears a little bit, not move away from oscillations, but talk more about the very unique properties of recurrently coupled oscillator networks. Because I believe that novel computational options have been provided by the implementation of such recurrent networks, which in evolution are fairly recent. They have been developed early in motor systems as pattern generators, but in cognitive systems, they are rather recent. A particularly impressive example of the implementation of this principle is, of course, the cerebral cortex. And I believe that the computational principles that can be exploited in recurrently networks do actually provide elegant solutions for problems that are still quite difficult to solve with the conventional feed-forward, even the deep convolution networks. And you will see in a minute why. Some of these principles are actually exploited in artificial intelligence solutions. They are called reservoir computing or liquid state machines or echo state machines, and proponents of this direction of research is Wolfgang Maas, Joachim Jäger, Warnemo, Mirasso, and Fischer. These people use these nonlinear dynamical systems in order to perform computations. So what is so special about the dynamics of recurrent networks? First, they provide an extremely high-dimensional coding space because the dynamics of these systems is so exceedingly complex. Mind you that even the three pendula that are coupled, the three problem, are analytically very difficult to treat, and if you have a large number of coupled oscillators, it really gets complex. This permits, at least in theory, the storage of vast amounts of information because one has a very high-dimensional space for coding available. It also permits, in principle, for parallel search and very fast readout, which is something that one needs. If you consider that if you are asked a question like, have you ever heard about this, you can immediately answer and say no, which means you have explored your space on the fly within a fraction of a second. Likewise, as we heard this morning from Pascal, you make a saccadic eye movement every 250 milliseconds, so 4 saccades in a second, which requires to read out from the system the priors that you need after fixation has started in order to interpret the sensory evidence that is coming in. And this readout and matching has to be accomplished every 200, 300 milliseconds, which requires very, very fast access to stored priors and matching operations. And I would suggest this can be done on the basis of such systems. Because of their high dimensionality, they allow for good separation of representations for later classification. This is trivial. And recurrent networks exhibit fading memory due to reverberation. And this permits the encoding of sequences, which is also something that one would like to have. So the hypothesis that I want to propose and then substantiate with data is that these properties may actually be exploited by the cerebral cortex. First, for the ultra-fast access to a vast amount of memories, and then for the very fast evaluation of relations between incoming sensory evidence and the stored knowledge, the priors, the stored knowledge that resides in these weight distributions of the tangential lateral connections, which is an essential prerequisite for predictive coding. It must be done every 250 milliseconds if one explores a visual scene. Then I would like to propose that this high dimensionality is used for the orthogonalization of representations, to push representations as much apart as possible so that they don't get superposed, and this should facilitate classification. And that it serves the encoding and storage of sequences because of this fading memory property. So, there are a few predictions that follow from these hypotheses that can be tested. One prediction is that the resting activity of the system should be very high dimensional and should exhibit a correlation structure that reflects the dynamic superposition of all the priors. So all the knowledge that this system contains and has implemented in the weight distributions of the recurrent connections is latently there, and the expression of this presence superimposition is the high dimensionality of spontaneous activity. If stimuli become available, if sensory evidence becomes available, that matches some of these priors, the system dynamics should collapse into a lower dimensional state. There should be substates generated that have a lower dimensionality that can then be classified. They could consist of specific synchronization patterns, and I will provide you some evidence that gamma comes in again here. And these substates would then be considered equivalent with a sparse code of a confirmed prediction, the result of a Bayesian matching. These result states, and I will provide you with some evidence, should be particularly good states for the induction of synaptic modifications, for the updating of priors, and they should be propagated forward preferentially because they have a fairly high degree of synchrony, brief bouts of synchronous discharges that will impact on subsequent processing stages. So let's see how far we get. For the testing of these predictions, one does of course have to record with more than one electrode. One has to exploit the relational activity patterns in the cortex. So multi-site recordings. One needs to present sequences of stimuli in order to investigate the possibility of superimposition of information. And one has to find a way to retrieve stimulus-specific information in high-dimensional response vectors, and this can only be done with machine learning. We started with this in cat visual cortex, recording with silicon probe number of neurons, getting these high-dimensional response vectors, and having a bunch of superimposed receptive fields, and then presented as stimuli, in this case, letters and numbers. So completely irrelevant for cats. Convolved these time series with a sawtooth function that mimic more or less EPSPs in order to get continuous functions, which just makes it easier to use support vector machines for classification. Then train the support vector machine that at a particular time after presentation of the stimulus, that the first stimulus was, for example, an A. And then tested later on with these trained support vector machines whether they were able to classify activity evoked in test sets. And it turned out, yes, it works. And the first surprising finding was that even though the stimulus was only 100 milliseconds, we were able to classify and to decide whether the first stimulus, this stimulus was an A or a D or whatever number we had shown over up to 1 second. So the information was continuously present in the network, probably through reverberation. We also found that if we presented sequences, an A and B and C, and had the classifier find out whether the first stimulus was an A or a D, we could still find out after, in the responses to the second stimulus. So the information about the first stimulus persisted across the responses evoked by the second stimulus, and slightly into the responses to the third stimulus. We could still classify what the first stimulus was. So information about stimuli can be superimposed in time in this network. And we could, at a particular moment in time, find out what the first stimulus was or what the second stimulus was in the right sequence order. So the network was able, even though we had just used this linear classifier, to accomplish an XOR classification. So there must have been some nonlinearity in the network that allowed to the generation of activity pattern that we then could classify with a linear decoder. So in summary, this experiment showed information can be is distributed across many neurons because we could retrieve it only by analyzing at least 30 neurons. They were randomly selected. 50 neurons were better than 30, but below 30, classification became difficult. That there is fading memory in this network. Information can persist across stimuli. It can be superimposed, and then one can decode from at a particular moment in time what the sequence of the stimuli was. Sequence order is preserved, and this is compatible with the predictions that one would derive if computations profited from the high-dimensional dynamics of recurrent networks. So one can derive a number of other predictions. One is that if these stimulus-specific substates self-organize are the result of a match between incoming sensory evidence and the information that is resident in the weight distributions of the recurrent collaterals, then it should take some time until classifiability becomes good, because one has the combination of incoming evidence information and the resident information. And if there is some unsupervised learning in this network, which is also a prediction that one would like to have confirmed, if frequent presentation of particular stimuli leads to an updating of the weight distributions in favor of this stimulus, an updating of the prior, then if one takes late trials in an experiment after the animal has seen thousands of these patterns, classification should become better than in early phases of the experiment where these priors are not yet so consolidated. Now, this can be easily tested. Here are the rate responses of the cells responding to such stimuli. So I said 100 millisecond stimulus duration. You have these phasic responses, but then there is this reverberating activity that is generated by the network itself. It's a mixture of off responses and a mixture of responses emerging from this recurrency. Then we sorted the trials into early trials in the experiment where the animal had seen the individual stimuli only 100 times, the late phase of the experiment where it had been seen 1,000 times, and looked for the classification ability of, in this case was a Bayesian classifier, of the stimulus identity. And there are 2 points to be remarked here. This is stimulus classification. So stimulus is offset already here. And the red and the blue are the classification performance. You see that the classification performance gets better way after the stimulus has terminated. It's mainly in this reverberating activity that classification is good, not in the primary responses. And that classification in all the experiments is better for the late trials than for the early trials, suggesting that this frequent stimulus presentation, the experience with these stimuli has, in a non-supervised way, changed the network in a sense that made it able to converge towards a state that is better to classify. The reason why classification got better with time was that if one looked at the principal components here, we looked at the first two components of this high-dimensional vector. They segregate after this offset in this reverberating activity, and they are quite well segregated at 300 milliseconds, can be then nicely classified. This is here for 3 different stimuli. But the segregation even increases all the way down to 700 milliseconds. And this segregation is faster in the late trials, where the priors have already adapted themselves to these stimuli, where the stimuli are already familiar to the network. The information contributed by these priors seems to reside in the correlation structure of the self-generated activity. Because if one shuffles the trial, which leaves one only with stimulus-induced correlations, then classification is much worse. This is after trial shuffling, then classification or with real-time relations, saying that it is not just stimulus-induced rate changes that matter here, but it is activity patterns that are self-organizing within the network after the stimulus has been switched off. So the conclusions here are: the network self-organizes into classifiable stimulus-specific substates within a few hundred milliseconds. The overlap of substates in PC space decreases with repeated stimulation, which makes classification easier. So it seems like a non-supervised updating of priors. And stimulus-specific information is contained in the self-generated correlation structure of these high-dimensional vectors. Another prediction is that if indeed priors are acquired by perceptual learning in this non-supervised way and are stored in the synaptic weight distributions of the recurrent network, then they should be present in the covariant structure of resting activity. They should somehow show up. And the question is, can one retrieve them? And the answer is yes, it's possible. In this case, we trained together with Andrea Lazar an array of readout neurons, 12 by 12, this time to reproduce the pixel brightness value of the presented stimuli. This was for our convenience because it made it easier to scan the activity and see whether there was a replay of stimulus-specific information. So if we presented stimulus F, this is what the reconstruction after the classifier had been trained reproduced. And then we used these trained classifiers to screen ongoing activity while the animal was doing nothing. And to our joy, we found that indeed stimuli that had often been seen by the animal were replayed within brief bouts of spontaneous activity. So these high-dimensional vectors somehow replayed what the animal had seen before so that we could detect it. Sometimes it played something that we could not understand, and we tested every now and then whether the decoders really did what we expected to do them. If we showed the letter C, the decoder showed that resembled the letter C. So this suggests that stimulus-specific relations are somehow stored in the substates of this high-dimensional activity. They are represented there, and they can be retrieved. They can be retrieved in resting activity. Another prediction is that if the statistical relationships of natural environments are stored in the weight distribution of the recurrent connections, and we assume that an adult, mature animal has stored all these statistical relationships that are constitutive for natural images, then natural scenes should induce correlation patterns that contain more information than those induced by scrambled scenes, because the stored information can be matched with the incoming activity. And if the stored information corresponds to what is corresponding to sensory evidence, then this is what follows. What one should see in this case is that there should be better classifiability of natural images than of their phase-scrambled counterparts. Phase-scrambled counterparts have the same energy, but they no longer comply with the Gestalt rules of natural scenes. Gestalt rules that we think are stored in the network, because these animals have some experience already. So we produced natural images, a set of 6, their phase-scrambled counterparts, recorded, again, massive parallel single unit and multi-unit activity from monkey visual cortex V1, have the receptive fields, have these high-dimensional time series, and then did the same thing as before, see whether a classifier, in this case was again a Bayesian classifier, could retrieve from these high-dimensional what the stimulus was. And it turned out that it was indeed much easier to classify natural images than their phase-scrambled counterparts. And this was not due to changes in signal-to-noise ratio or changes in spike rate, because the firing rates, different pictures, were exactly the same. They were virtually precisely overlapping, and also the reduction of the Fano factor, of the variability of the responses that inevitably occurs if one stimulates the cortical network, was exactly the same in the two cases. So this is not a signal-to-noise thing. There is more structure in the vectors or trajectories if the sensory evidence complies with the stored priors. We then looked, it's a gamma thing. What happened with the oscillatory activity, and to our great satisfaction we saw, because there's a prediction that the better sensory evidence matches the stored priors, the lower dimensional should be the resulting matching state. And this low dimensionality should be reflected by more coherence, by better oscillations. And Martin Fink has written a nice review last year saying that the better the fit, the more gamma you should have. Now this is what we saw here. We see more gamma, this is gamma power here, in responses to natural scenes, then we see it to these phase-scrambled counterparts. There's a ramp-up of gamma, as you can see, because these experiments were done in the context of attention tasks, and the monkey was expecting a cue to occur here, which would tell him which of the various pictures he should attend and respond to. So this ramp-up is essentially due to expectancy. It's a hazard function. It is stronger for the natural images than for the phase-scrambled images. And we were, of course, curious to see whether we could also decode stimulus identity just by using gamma amplitude from these various electrodes, just using this time gamma power and not spike rates and coincidences of spikes. And yes, indeed, one can decode quite successfully 60% of stimulus identity simply by taking gamma, and it works better, as expected, for natural scenes than the phase-scrambled scenes. Again, because more information available, which is good news, because for brain-machine interfaces, field potentials, they can always be recorded from. And if classification with 60 neurons or 32 electrodes is already producing a 60% classification success with fairly complex natural images. One could use probably just few potentials and get away with them. So classification of scenes is possible, both from spike and LFP vectors. It's better for natural than scrambled scenes. It's not due to changes in signal-to-noise ratio. It must have to do with internal structure, correlation structure of the resulting matching result. And natural scenes induce stronger gamma. We would predict they produce a stronger reduction of dimensionality. We are about to analyze dimensionality. It's a bit tricky. Maybe. So the proposal is that the match between natural scenes and stored priors leads to well-organized substates that are reflected by a gamma ramp-up, and that is associated with better classification. Now there's a last prediction, at least for which we have evidence. There are many more which need to be worked out. And this is that states representing good matches, so low dimensionality, high coherence, probably strong gamma, they should also be favorable conditions for induction of synaptic plasticity. And they should lead to an automatic updating of priors. Why so? Because the synchronicity that goes along with gamma oscillations, but could also be theta or any other frequency range, but the higher the frequency, the better the precision and the cooperativity, is in postsynaptic depolarization, and thereby favors the surge of calcium through NMDA receptors and voltage-gated calcium channels, what one needs in order to induce synaptic plasticity. Here's how it goes if two cell populations are well synchronized. Pascal talked a lot about it this morning. The synaptic potentials of the EPSP is produced by A impinge B when it is just at the depolarization peak. So NMDA channels can open, calcium can enter, and so forth. So it's a favorable condition for the induction of synaptic plasticity. And we wondered whether one could find some evidence for it. And as a paradigm, we used the induction of changes in orientation maps in the visual cortex with 1 hour of continuous stimulation with a drifting grating of a particular orientation. This induces changes in the orientation map. And at the same time, we recorded with floating electrodes single units, experiments done together with Ralf Kaluske. And the change in the map is due to the fact that neurons that are still able to respond to the inducing grating minus 30-degree preference offset, they shift their orientation preference towards the conditioning stimulus. While neurons that are far off with their tuning, they don't move or they become less responsive. And this one can see if one compares these maps carefully. Here, 45 degrees was conditioned, and you see there's just an increase of readiness here. And here you see neurons that have an initial tuning. Here we conditioned the zero orientation, an initial tuning that still allows them to respond. They move up and start to respond very well to the conditioning stimulus and a little bit less to off stimuli. Now, this was a serendipitous finding because we saw variability. It didn't always work. And we wondered, why does it sometimes work and why does it sometimes not work? And so we went back to look at all these conditioning trials, and we found that the variable that distinguished the successful conditioning experiments from those that were not successful, and rather produced the inverse, a reduction, a habituation of the conditioned orientation, was the amount of gamma evoked by the conditioning stimulus. If there was, during the conditioning phase, strong gamma, everything else unchanged, there were these fairly drastic changes of orientation preference in neurons that were able to respond to the conditioning stimulus, while nothing significant happened in all those trials where the gamma, induced gamma, was absent or weak. So we think that these changes have occurred at the level of the intrinsic tangential connections and not at the level of the input connections. because we saw these changes in the maps also after whole field conditioning when we just evoked the maps with a small patch of grating. This is the map evoked with a whole field stimulus. In this case, the map is emerging nearly the same map when we stimulated only this small patch of cortex. This activity comes from the tangential intercortical connections. So it's a subthreshold activity. There is no direct activation of these neurons. But the map changes are also seen out here with this local test stimulus, suggesting that the changes must have occurred at the level of the intrinsic connections. So this brings me to the end. It appears as if synchronous gamma oscillations would favor the induction of synaptic modification. This is why I was so excited when I heard Earl's talk this morning, where he said that it was these gamma bubbles that may have induced a brief change in synaptic efficacy. And these changes occur at the level of intrinsic connections. The same that we want to change for the updating of priors in a non-supervised learning. So, in conclusion, gamma oscillations seem to reflect the match between evidence and priors. They reflect constrained substates with specific correlation structure. Well, I didn't show you any evidence on that. And they seem to favor Hebbian synaptic modifications. No evidence provided today for the enhanced transmission, but Pascal showed you all the necessary data for that. Now, let me end with a speculative hypothesis. Seems to me, at least, that with the evolution of the cerebral cortex and the hippocampus, a novel principle of information processing emerged which capitalizes on the prevalence of reciprocal connections and massive recurrent feedback connections, which you don't see in insect nervous systems or in mollusks, except for pattern generator circuits. Much of this is based on the exceeding complex nonlinear dynamics that such networks exhibit. And this provides the basis for computations in high-dimensional dynamic space. And there are many more possibilities opened by this scenario. So the hypothesis is that with the evolution of hippocampus and neocortex, a new computational and very powerful computational principle has been exploited or is exploited. And I believe that this is the reason for the tremendous evolutionary success of this structure. Because since it had been discovered by evolution, nothing new has been invented. It's just more of the same all the time. And this despite of the susceptibility for epilepsy, because such networks come with a high price, and this is of course instability. They can only work in a very, very narrow range of excitability below epilepsy threshold and above falling dead. So they have to be regulated in a very subtle way in order to be functional. Okay, and these are the people whom I owe all the thanks. Andrea Lazar, who cooperated a lot on this reservoir computing. Patrick Jedlica, when he did a lab rotation with us, worked on the improved classification of the presented stimuli. Danko did the experiments on fading memory in the very beginning. Gareth Bland has left us, unfortunately. He was very important in helping to record and sort data. Liane Klein recording a lot of this data. Johanna Klonlipok, my technical assistant, who can do all this work by herself. Thank you very much.